Hello AP Biology students, we're going to go over the fourth section in Unit 6. And this is going to be a short section that's going to look at the central dogma of molecular biology. Remember that we're focusing on DNA in this unit, and DNA is the genetic code of life. That pretty much means it has the instructions to determine an organism's characteristics or its phenotype. The central dogma basically explains how DNA then codes for these characteristics. Or in other words, just going from DNA to RNA and from RNA to a protein. Because remember, these proteins are going to give us our actual characteristics. The first part of the central dogma is replication. We've already gone over that. That's when DNA copies itself. The second main part of the central dogma is transcription. This is when a sequence of DNA is going to be copied into something called mRNA. The last or third part of the central dogma is translation, when mRNA is read by a ribosome and converted into a protein. The mRNA specifically isn't going to be made into a protein, it's just the genetic code or the copy of the DNA that's going to be read to make the protein. Now, when talking about the central dogma, we have to know a lot about this molecule called RNA. RNA stands for ribonucleic acid, which means that ribose is going to be the sugar instead of deoxyribose. Uracil is also going to be used instead of thymine, so in all RNA, you're not going to see T or thymine, you're only going to see the uracil. However, the other nucleotides are still found in RNA, such as adenine, guanine, cytosine. Well, RNA in this case is just going to be looked at as being single-stranded. Yes, there is double-stranded RNA, but we're going to focus and just say for now it's going to be single-stranded. I want to make a quick note about the difference between RNA and DNA. There is a lot of functional differences and even structural differences, but when we look at ribose and deoxyribose, there's not that big of a difference. You can see here in the ribose sugar, we have an extra oxygen here, where in deoxyribose, we don't have that oxygen. I always felt when I was growing up, I thought of RNA being very, very different than DNA, and it is, but it's not really that different. Now, whenever we go from DNA to RNA, whenever we transcribe DNA into RNA, we're gonna still have that complementary base pairing. We're still going to see C's bonding with G's, G's bonding with C's, and T's bonding with A's. However, when we have A on the DNA side, on the RNA side, you're going to bond it with U or uracil. This is again because, remember, RNA doesn't have thymine. There are three main types of RNA that we're going to talk about in the next couple sections. We have mRNA, tRNA, and rRNA. mRNA stands for messenger RNA, and this is just a copy of the DNA that's transferred to ribosomes. mRNA typically looks like this, where it's just a long strand of RNA nucleotides. tRNA, on the other hand, is called transfer RNA, and it transfers amino acids into the ribosomes. It obviously kind of looks like this T, and you can see here, it's all going to be made of a single strand of RNA with an amino acid attached. We're going to talk more about tRNA and how they're involved in translation. And lastly, we have rRNA, which stands for ribosomal RNA, and it's basically part of the ribosome. So ribosomes are made of two things. They're made of proteins, they're made of rRNA, and a lot of students forget what rRNA stands for or even does. It just helps make up the ribosome. I saw this meme a couple months ago and I thought I have to use it in my PowerPoints because I absolutely love it. You can see the flow of genetic information going from DNA to RNA to a protein. Perfect.